let's wait a sec. 56, 45, 36.
It's really hard to tell if I'm actually touching the ground.
I know. 39.
I think I am touching the ground.
Oh, really?
I think
Yeah.
I can zoom in a little closer.
Sure.
But that Van Gogh is really tough to see past.
There, I'm not touching the ground now. I saw it come away.
Okay.
So if that works, that'll be filter safe.
I'm at 18.
Ah! Can't
I
win for
know, a
losing.
centimeter makes a huge difference. 20.
Baffling.
14, 10. $16. $13. 9. Yeah, so we need a little bit warmer again.
Okay.
Can you move in half centimeters?
I mean, yeah, 1.5.
You know, if you want to come wide.
Back to the left, maybe just a hair, and then try it down a little bit.
There?
Yeah, that looks good.
Okay.
Let's see what
I would call that a 0.5 centimeter.
Yeah. It's at 6.
Ooh. Ooh.
Hold on, though.
Holding.
Might change. 12. What are you getting on the thermocouple? 10?
10.
Yeah, I'm at 11. 3. Julie, do you think this is the same spot we were in?
Can you come out a bit, Jess? We'll take
I
it.
can't tell.
Yeah, I thought it was further down, but I could have been wrong.
Keep going
No,
out.
that might be.
I think it is. I think it's really
It is.
hard. Yeah, I
Yeah.
think what's happening is, I think, because
Such a
we're getting
small
such a lot
spot.
of diffuse flow in the field of view from the camera, but actually when it clears, I think the wand might actually be set just behind it.
Uh-huh.
So
I did knock the rock from when we first had
Yeah.
watch change, just to mess with everyone. So
I don't know, sometimes
I'm
it might
only
be,
at 10.
if one of you want to come and stare at, when we get a zoom in again,
go ahead and zoom.
I don't know if one of you want to come and look at this screen, just because it's a bit
Say what?
closer.
Go
Yeah,
ahead.
Go ahead, I'll watch the temperature.
Okay.
Yeah, it's just when we get those moments of clarity, can you see, like, I think it's in front. I don't know if that...
You're thinking I should bring the wand towards the vehicle?
I think so.
I think so, yeah. Just at
Sure.
at a
A scotch?
point, yeah, 0.5 of a centimeter, like a tiny, tiny amount.
Not like that.
All right, now it's at 40. Temperature's at 45. Yeah. 47, 52. Okay, that, yeah. Nicely. Sweet.
Good
Okay,
call.
that's 36.
Oh.
35.
There.
Hold on.
Can't count those eggs. Chickens haven't hatched.
35. It looks like it's... Yeah, it looks... Can you see? It looks
steady
like it's
at, so now we're at 28. It is steady though.
All right, 20.
Oh.
It's okay. It's all right. It's not your fault. 29. What do you think, Julie?
For it.
All right. Lock and load.
All right, arms
All right,
locked out.
Restarting the clock for 20 minutes.
Okay, hold on. I got to get port
Yeah.
1.
Okay, we're standing by. Would you like the bubble on the flow meter?
Yes, please. Moving to port 1.
Port 1.
Temperature's at 27, 22.
20 degrees. Okay, at port position 1. Starting filter slow.
Okay, starting at 1915.
Go, wheel, go. I'm using my mind to move it.
Hey, Jess,
We're all willing it forward.
Yes.
Come on, paddle wheel.
Go,
yours?
Temperature's dropping a bit.
It's 18.
Go ahead, lounge.
Down to 15.
I was going to say, it now looks like when we had that moment where we didn't have the diffuse flow in front of the screen, it looks like it's behind the probe again.
Behind
so
the
I think
probe,
it's,
yeah.
it's just very slightly
Yeah.
just
It can go
like
down a little bit. I mean, yeah, I'll leave that up to you.
We're right about 12, so let me pause for 1 second.
Are you pausing sample.
Okay, yeah, so if you can move, see if you can get,
Maybe one scotch down.
Back in there.
Okay.
You're still at 1.
it's okay,
Oh, okay.
just for a second.
Okay.
That was a little too far. Let's try that.
27, 32, 39.
That's it.
There we go.
Yeah,
That's the
48,
ticket.
that's the ticket.
63, 64,
Oh, oh, no.
Is that too much to ticket?
74, 80,
80.
What's our max?
50,
That's
95.
more
Oh,
than
move it,
50.
move it, move
Oh,
it.
yeah, move it.
Don't want to burn it. 111.
Continue toasting it. All right.
69. 62.
Let's try right here.
82.
That's not a good spot. Let me think. Try that one again.
Well,
It's right around 63
Yeah.
right now, so
Oh,
just,
too much.
yeah. 101, 111,
oh, move it.
127.
Oh.
Bail out, bail out. Yikes, this is touchy.
Yeah. 62.
I'm slightly left of where I want to be, I think, but
55. 51.
I think it's going to heat up as I drift back towards it.
Yeah. 64. 55. 47. 56. 63. 57.
How strict is that 50-degree max?
I'll leave that up to you, Sarah. Nobody can hear you.
Was the 50 degree the maximum on the IGTs? What was the
No.
min and max for the IGTs?
No, but I think it's more for your microbes.
Oh. I mean, it doesn't look like it's going to be
I'm kind of stuffing in the
60
muck
the
here.
whole time.
Where are you at right now?
It's at 60. Well, now
At
it's at
70.
70.
I'm kind of I kind of drifted
Oh,
down.
now it's at
okay.
85 and rising.
It was pretty steady around 60.
It's a very, very slight vehicle movement that's doing it. We're like
Yeah.
kind of on the edge of a shelf cliff thing, so it's not exactly the most
Right.
deluxe parking spot.
We're at 65 right now.
I think in the interest of time, we have to accept it's going to vary from, 15 to 100, and that's okay.
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 It's pretty steady around 65 right now, so you're okay for go?
Yes.
Okay. Starting the clock.
And if it goes crazy, if it goes, plus 100 or sub 15, then we'll pause, move it again, and line up and do it again, I
Okay.
guess. I'm fine with that if you are.
Okay.
That's fine.
We're going down to 50 now.
Yeah.
Holding steady around 60. $66.
Hey, Sarah.
Yeah, what's up?
She's here with me. Do you want to talk to her about your affiliate?
I can't hear you that well.
50.
Can you hear me now?
Sorry.
Yes.
I was just saying that Roxy is here, and she was
Oh, really?
asking if we'd gotten any of the purple stuff.
Hi, Roxy. Yeah, I think after this sampling, we're going to look around in this general area to pick up a rock. I found a lot of them were settling on some other worms kind of things, which I've noticed in some previous papers that they have been known to do. It was kind of hard to tell how much biomass was on there, though, especially when they weren't submerged in any seawater.
Roxy was just saying that they got some at some seeps in Guaymas, and they were attached to worms also.
Yeah, yeah. So today, if I'm looking for some ciliate mat again, I'm going to look for a rock, and then, I don't know, the last rock had like so much other biomass on it, it was really hard to differentiate when it was outside of water. So I also will maybe like submerge it in some water on chip, or try to get like a better zoom in before we put it in the bio box here. Temperature's at 28, by the way.
Okay. That's a good temperature, too. Do you see from the colonization chambers you've brought up, like, are you guys seeing stuff on the chitin the tube worm shell stuff that you
Yeah,
put down?
I definitely see like some growth on the Riftia bits that I put in, but it's just like little black dots. I don't know what it is. I'm excited to, I don't know, figure that out. Temperature's at 30.
Since we're just sitting here and I have you, the
Is it
Niskins
sorry,
yesterday,
one second. Is it possible to get the volume up from ISC at all? Hello?
Yeah, stand by. We'll ask video. Video.
Video. Is there possible to get volume up from ISC? Is a little bit? Okay, that's fine. All right, that's fine. Go ahead, Julie.
Talk louder.
Okay. I just turned my volume up really high. Go ahead.
The Niskins that you guys got last night, the deep and the shallow, did you filter those?
Yes, I filtered. I didn't filter the background seawater because I'm only filtering those for when we're taking samples. And then I did filter the 150 meter sample.
Awesome.
Yeah. It was super fun. I love filtering water. Temperature's at 32. Yeah, so my goal with the ciliate rock or sample today, I want to find a piece that has maybe a little bit more blue ciliate mat than previous and not as much biomass on the other stuff. When we collected the other sample, it was thrown in the bio box with microcolonizer and a couple other rocks that also had biology on it. And so maybe that's why it was so covered, but yeah, we'll try again today.
Just a really quick note, Sarah. I don't know if you've noticed the paddle wheels pretty lethargic.
Yeah. That's not necessarily a perfect indicator that, like, if it's not moving, it doesn't mean that there's no water being filtered.
Okay.
Especially with these filters, and what we found yesterday was this was kind of how the first site went, and at the second site it moved a little faster after we had done a bag and some other stuff, so maybe it's
pushing
moving
air through the system or
maybe,
something?
but we are filtering onto 0.2 micron filters, and so it's kind of to be expected that maybe
A bit slow.
it would be a bit slow, yeah.
A bit of a slow start.
Yeah.
No
hasn't
worries,
had
I'll just
hasn't had
keep
any
an eye on
coffee
it.
yet.
Yeah.
Temperatures at 34.
Copy that.
Roxy came here to watch the exciting world of super sampling.
Yay.
She's out of here.
And gone.
Do I want to know why there's an entire box of Snickers next to the station?
Yes, you do want to know. The answer is Susan.
Susan's
stash.
fantastic. Wow, look at that.
She is
Snicker
the
Susan.
hero
She
of
is.
our
Her
watch.
personal stash? Or, okay, she's sharing it? That's
Yes.
great. Wow.
No, she doesn't share. She has a 30-pack without us.
Gallon of Snickers just for her.
Airplane with that,
Wow.
and a big thing of M&Ms.
I'm surprised she must have bought them in the airport.
No, she said she had it in her carry-on bag, and it was really heavy.
Wow.
That's commitment.
Such
Incredible.
preparation.
And she doesn't even like Snickers.
Wow.
True hero.
True hero.
That's the best way to have willpower, is just buy a snack you don't like to share.
I don't think I have that kind of willpower.
I don't have that kind of snack.
Can we get a temperature update, Sarah, please?
40.
40. Perfect. Thank you.
So did they cut out some of the material from the exterior of the Super yesterday after it? Oh, no. I think it increased the efficiency of it, you know. Hydrodynamic. Fluid dynamic, yeah.
Temperatures at 52.
So are we looking for a particular temperature before continuing on? Or
We're continuing.
continuing?
Yeah, we're filtering now.
Okay. Oh, I see the spinny wheel. Okay.
Yeah.
We're keeping on, keeping on.
Excellent. Did the hose get a little crispy yesterday?
Toasted.
Toasted.
It
Okay.
looks like
Yeah.
it's seen better days.
I don't know. I don't say that about my marshmallows. This is the best day they've seen.
It's the new style.
Yeah. Punk rock.
It's
Punk rock
aged.
super. Vintage.
Vintage.
Vintage sampler hose. What is the sampling wand made out of?
I know this. Eric had, oh, watch, I'm not going to be able to get it.
Oh, he was talking about the 3D printing.
Yeah. It's a,
You were there.
it's
I
a,
can't remember the material.
it's a type of polymer, right,
He,
that's been cured
I
at
think there
each
was
level.
like a couple acronyms he used.
Some letters.
He did.
I didn't,
Yeah,
get
some
the, I didn't
letters.
get the acronyms, but I know he used the word polymer. And
Oh, cool.
it's 3D printed, and each level of the 3D print is cured into place, so there's no bubbles within it, which is supposed to help the stability and strength at depth. Yeah, that's as much as I got.
It's pretty good.
We'll have
Okay.
to get Eric in for the
Got some more details.
full detail.
Temperature is at 41.
I think it's on the internal side. not on the remote side. Power cycle.
Power cycle, the
Power
robust
cycle.
term.
Well, you were having trouble with the IPOD beforehand too, weren't you? Yeah?
Yeah.
It's device independent, I think.
Yeah.
Did you guys isolate the white filamentous looking bacteria maybe off the para-alvanella?
Yeah, we
Yeah.
did separate it. I don't know what we're going to do with it, but we did separate it. Maybe a bacterial mat of some kind.
You
or
think so, yeah.
Oh,
Because they
not sure.
can get that filamentous shape. And it was just interesting because it aggregated on like one solitary
Yeah.
tube
Yeah.
versus
Just on one
others.
of the ridge
Yeah.
We've seen them.
Oh,
We've
that's
seen
what it
them
was,
off and
Ricky?
on though, right on the
The white filamentous.
yeah the right
bacteria.
filamentous.
Yeah,
But there's
there
this whole colony and
yeah,
only
there was
one is...
one rock that you sampled,
Right.
that we sampled that had one ridge deer, and then
Yeah.
it was just like, yeah, super, super fluffy. But as you say, when it came to the surface, like trying to
It's
to
really
actually
hard.
visualize it was really difficult.
know.
But
Really?
subsea, it looked incredible. It looked like the whole thing was really fluffy.
And then it
Yeah.
just collapsed.
yeah,
Yeah.
kind of just tend to.
That's kind of why I think tonight when this comes up, maybe we should submerge stuff in seawater or in the
Seawater.
bio box. And
yeah, we can take some
grab
water
some
out
of
the
that water.
biobox.
Yeah, I'm not sure.
So Sarah, how did your sample processing go last night?
This morning?
This morning, yes.
What day is it?
Exactly.
It was this morning.
It was this
Where
It was
am
this
I?
morning.
Who am I?
Nav? No.
Yeah.
So, yeah, it went fine. It went well. I grabbed them 2 microcolonizers. They're covered in limpets. So I didn't save any of them. But they were definitely about 1 million based on my count.
About 1 million?
About 1
How does
million.
one calculate that?
1, 2, 3, 4 million.
And then, thank you. And then, yeah, the inside tubes are looking really good, meaning that I've seen on the 2 substrates we have are shell and riftia tube that have been sterilized and have been inside those tubes or those cones, and I see like little dots on them, which means that something's growing on them. I assume other microbes have also settled on the different rocks and substrates we have. So we separate that out into 2, one for SEM and microscopy, and then another tube for DNA analysis. So we'll extract DNA to kind of get an idea of what settles on those things.
Awesome.
But it went okay.
So how long did that entire process take for you this morning?
I don't know. I had another time point from a previous experiment because we're doing incubation experiments this morning, and then I was also filtering some seawater that we collected from 150 meters as like a background near surface sample. So I think that took me about 3 hours this morning.
Wow. Okay.
Total. Temperature's at 58.
So the 2, excuse me, the 2 colonizer experiments that you collected last night and processed this morning, you said had shell and tube worm casing as the substrate.
Yeah.
What about some of the other substrates that we had seen before the cruise started?
Actually, all of the microcolonizer experiment, those cones have the same things in them.
Okay.
Yeah, so there's like 6 different slots for tubes to go. Let's see, it's shell, riftia, quartz, pyrite, basalt, and olivine. And so they're all, we're getting like 2 replicates essentially every time we bring them up. And I assume that there'll be more growth and more limpets after we pick them up today, the last 2 today, than the last 2 days. So kind of a time course.
And over 1 million limpets, not
exactly.
over, in just a few days since those colonizers were placed.
Yeah, I mean, I think on our second dive, we passed over that same site, Mount Edwards, and they were also covered in limpets. So it took less than 24 hours, I think, for those limpets to kind of take over.
Wow. That's impressive.
Yeah, let me know if anyone wants them. I'll have more today.
Okay. Was Megan and the data logging team?
Yeah, I
Can you
think
get any of those?
she did
Yep.
grab a few.
Okay.
Yeah.
Great.
kept offering her more, but she didn't want them. I don't know why.
1 million limpets.
We've already got a big jar full of them in the fridge.
Oh, I know. If I just add them without her knowing, do you think that'll be?
Oh, so viewers are curious about, you know, what happens to samples when they come up, and particularly if they smell when they come up, since we know that there are a lot.
Oh my goodness.
Yeah. Tell us more.
Yes, they do. We have a very sulfury smell when we have this vent fluid. So the labs smell wonderful for the few hours that we're sampling them. So yes.
Yikes. Yeah, it's, as soon as the vehicles come back on deck, you know, even standing a few feet away, you can definitely smell something.
Yeah, and it's very, so we're filtering on filters right now, but then we use another wand that's similar to this from the same site, and we'll fill bags. And the last couple bags that we've brought up have also been they're like gas tight and pressurized, and so we usually use a pump to pump fluid out of bags or whatever container we've sampled something in to go through filters and into our incubation experiments. But as soon as we hook up that tubing, it just starts going and smells like sulfur.
Yeah. And concentrated, right? Because it's being filtered out?
No, the bags are just the fluid collected.
Oh, okay.
Yeah.
Got it. Yesterday, though, when the vehicle, or nope, it wasn't yesterday, it was this morning, 4 a.m., when the vehicles were back on deck, it smelled like corn checks. It was the strangest thing.
I did not have that smell.
You
I
repeat
did
that?
not experience
I
that.
did not
Corn
see
what?
that. I did not observe that.
I guess I should say corn cereal, not a sponsor. Yeah.
We did not share that experience.
No, it was like right when they came back on deck, and it was before Gabby started hosing down Hercules.
Wow, just
Corn checks.
Corn checks.
Interesting.
I don't know what that's diagnostic
Temperatures
of.
at 51.
So like the moment you guys pulled it out of the
Copy
water.
that.
Just
Yeah.
corn cereal.
Corn cereal.
Someone had an early breakfast.
Yeah. It's very strange, because every other day it's been very sulfurous and smells like, you know.
Well, we did do that
Eggs.
geotransit
Refreshing.
that was in totally different, I guess.
Maybe that's
more
what the high
corn-filled
temperature vents
areas.
smell like.
There you go.
Yeah. Yeah, so we've been, I think the low we saw was 38 while you were gone, recorded temperatures. Yeah, and we're at almost 18 minutes filtering, so we have a couple minutes to go. I gotcha. Here you are.
And Sarah, are any of the traffic cones or colonizer experiments
Colonizer experiments.
staying down for a longer time, or will they all picked up before will they all be picked up before we leave?
This dive will be picking up the last two.
Awesome.
Yeah.
Okay.
I can't leave them behind.
What was the
Yeah.
process to make them? Like,
So this is a kind of a collaboration with Pete Gerges' lab at Harvard, and so they came partly assembled, and he's deployed them previously on a couple of dives or a couple of expeditions, and I think that they have been down for up to like 1 or 2 years, but I'm not sure yet. Other microcolonizer experiments that are very similar have been down for up to multiple years. But there's a lot of titanium wire, zip ties, and electrical tape involved.
okay, a little mix of unusual materials and off the shelf.
I was going to say, as with all oceanographic experiments.
Zip ties,
Yep.
electrical tape,
I'm-
wire.
I'm a real fan of science that uses electrical tape and zip ties.
Absolutely.
It's a common man's material.
Reliable.
Before we end this sample,
trustworthy.
Very
can
wasteful.
before we end the sample, can we zoom out a little bit so we get a comparison shot?
Okay. Yeah. And we're at,
30 seconds.
We're
How
about to
far? That
finish.
far, Aubrey, or more?
this is at 20 minutes.
That's good. You can put it back in if you want now.
Okay. Ooh.
Okay, stopping.
Temperatures at 40.
Copy that, stopping sample.
Okay, we got 0.11 liters per minute.
0.11 liters
not
per
planning to.
minute.
We're doing
2.21 liters
more of these.
filtered.
2.21 liters filtered. Thanks, Amy.
Sure.
What about it?
I'm going to
That was
advance.
great. Look at it. Pinging away.
Temperature's at 58.
We'll do the second of 3 filters.
Just confirm we're moving to port 2.
Reach port 2. And begin flow.
Copy that. Starting sample 1940.
It's going fast.
That's really strange because it was still really quite slow up until the end of
Right.
that last sample.
Yeah. Might have been a big bubble.
So that really suggests there was like a, or a pinch point right in
Yeah.
the tubing, the filter one maybe.
Yeah.
Yeah.
Amy, just make sure you put that in your notes.
Okay.
Yeah, because now it's trucking along.
Yeah.
I feel like yesterday's dive, though, it was doing the same thing for all 3 filters on the first site. And then the second site, when we started filtering again, it was looking better like this. So I don't know what it is.
Temperature's 29.
You know, it's cool if you look at the biology right now, in the top corner by the probe, or not corner, to the top center, you've got one of those pink roly-poly things. I forget the scientific name.
Polynoid.
He's pretty big. And then if you look at the bottom of the screen, there are a couple others that are like a quarter of the size of that one. And we're at full zoom, I'm pretty sure. And so it's just wild to see how extreme the differences in their size can get.
Lee, do you have any fun facts about these critters?
Their scales fall off when they come to the surface. They're not so happy.
Really?
Aww.
Practical
Yeah.
insight.
Yes, practical fun
Practical
facts.
insights.
Have we brought any up?
Yeah, there have been some polynoids.
Yeah.
Yeah, so these are a type of scale one.
Do the scales fall off throughout their life, too?
To be honest, I don't know. I guess so. I would assume so. To a certain extent, they might lose a few, but yeah, they're very delicate when they come up, so the scales do float off. Especially when they've been in a sampling chamber with lots of limpets and things like that. I mean,
Yeah.
the abrasion, I think that causes
Yeah.
the scales.
Yeah, like if you look at the bottom middle of the screen right now, there's this very tiny one. And I wonder if that's just a juvenile or if they just vary that much in size.
I think it could be that they vary, but also that could be like the other side of it as well. So they look kind of iridescent on their underneath side. So that might be a slight angle.
Temperature's down to 11. Lee, there was what I thought was in the plume a whole bunch of shrimp just swimming around, all those little orange shrimp.
Oh, really?
Yeah,
Ooh.
like the ones we saw at Louis-He.
Oh, interesting.
Yeah.
When was that, this morning? Or
No, at this vent.
At this
In the
vent?
hot vent. Yeah, there's a big plume above the candelabra vent. And it looks like there's just millions of little shrimp.
Oh, interesting. I didn't see those
Yeah.
this morning.
Maybe when we finish, we can zoom out and see if that's what they really are.
Yeah, no,
I wonder if
absolutely.
the plume kind of creates like a little aggregation device, like a little front, you know,
Little
that may
vortex to
push
trap
out
them.
particulates along the edge of that plume.
It's
I
really
don't know.
interesting to see shrimp in the plume, though.
Yeah,
Yeah,
they were definitely
really,
swimming.
really interesting.
Yeah.
Oh, they were scavenging in the interior of the plume.
I don't know,
That's
but
cool.
it seems like they were swimming kind of around, and then they would get sucked up in the flow and go,
And then
and
come back in.
and then come back. Yeah, like they were
interesting.
on a ride.
That's always my favorite part when you see them come through the camera. Looks like they're at the mercy of the flow,
Yeah.
scuttering away, trying.
Well, I guess you don't have to invest any energetics in being buoyant in that part. We.
We.
I like the very scientific
Thank you.
description of that. That could also be summed up by we.
This is the watch of highly scientific terms.
Well, you know, you started out with like, oh, you don't need as much energetics for that. We.
That's how I form all my hypotheses.
Fantastic.
Super
In lay
temps at
terms.
17.
17, copy
I'd love
that.
to read your thesis.
Yeah.
Animals saying we.
What's that commercial?
So Amy,
Yes.
what is that flow rate there? That's about a liter per minute, right?
Let's see.
Or
I don't know how
0.7
to
or
calculate that, but it's because the flow rate that I have
Put
on
in
here is
numbers
counts per second.
here, right? You put 844
Okay.
and then
Hold on just a sec. We're trying to. Oops.
no, it's okay.
you put in like just
How many
a
minutes
17.
have we gone? Oops. I don't
No.
know.
6 minutes, 20 seconds so far.
Yeah, it's like 4 times as fast. 16. So we've already
Can you turn
filtered
power off to the
2.3
mezzo?
liters.
We'll let them maybe sit for 10 minutes or something.
So we
power
should
it
get
off.
about
Let
6
them rest.
or 7, or no, 8,
Yeah.
8-ish.
Yeah.
Let's
Yeah.
do math.
Yeah, we'll have a
That
look.
sounds about right. Yeah. Nice.
Yeah.
Yeah, I'm wondering if perhaps
Sure.
there's no filter.
Maybe
in
5
this
meters,
one.
210.
No.
It's going
Well,
so fast.
what?
I checked
No.
them.
Yeah,
I didn't
yeah,
check
yeah,
all
yeah.
of
No,
them,
we
but
checked.
I checked. I know I put filters in all of them,
Yeah,
and then I
yeah.
double checked 2 of them, and
See
they
what?
did have filters.
I mean,
Maybe there's a hole in it or something.
yeah, sure.
Yeah, I
but
mean, it being this fast,
You'll get rid of that by
there's
turning port.
a leak in a different way,
Yeah.
you know? But we'll have to see what number 3 looks like.
Okay.
does this site have a name?
Candelabra. But that's really the big vent, so we could.
Yeah?
It could be Phantom.
Oh no,
Phantom
I'm
of the Opera. Or Liberace.
Is candelabra, so is that what we're seeing in Argus now with the vent to the right of us?
Yes.
So that's actually candelabra.
That's pretty
like-
cool.
Ah, okay, cool.
That's a cool shot.
It is a cool shot, actually. It's a really neat little mound, actually, isn't it?
Yeah.
That we're
Or Lumiere.
Ah.
that'd be cute.
Yes.
But we are not putting out another marker here, just so you know. I mean, we can see the
Okay.
other marker. I'm sorry. So I suspect the reason your temperature is so much lower is because you're pumping that much faster.
Yeah. It's, yeah, around 20.
So is that a function of it in training more of the ambient water around the outside?
Yeah, I
Yeah.
think so.
Okay, understood.
But it doesn't look like there's as much wafting as there was at
There's
the
a little bit
beginning.
though.
There was definitely a lot more shimmer in front of the sight earlier.
Yeah, it's definitely a lot clearer to see the location of the end of your super probe in relation to where the flow's coming out of that little diffuser.
Yeah. Is that scale worm coming over to our event to have a drink?
I think so. Come to join the party.
Watch out.
Watch out.
It's going to be fashionably late.
Oh.
It's fun looking at the dive footage later on, and you'd fast forward it. You just watch those little guys scurry all around the screen.
Oh, really?
Yeah.
We're at 36 degrees.
Copy that.
Do they, are there other color or other morphologies, or is this pretty standard?
For the polynoids, for the scale worms?
Yes.
To be honest, they do look all very similar from camera subsea.
Okay.
Once you get them on the deck and you start looking at them, you look at like their bristles and their mouth parts and things, I'm sure they look very different to polychaete taxonomists. But yeah, the morphology is very similar and the colours are very similar in terms of that kind of pinky colour. Although that one has a really weird long pink stripe down
Right,
That's what I was
on
going to
the
ask.
dorsal.
Yeah.
back,
I was
which
wondering
is interesting.
I wonder if it's the angle at which you're looking at his scales. You're just watching, seeing like an overlay pattern.
Looks like a real feature to me.
It's
I
yeah,
don't know
that
if
is a bit
it's
strange.
below the scales or.
I don't know, this camera's really out of focus.
Sorry, it's the Van Gogh.
Video. Can you remove the Van Gogh filter, please?
The impressionist filter.
Hashtag no filter, guys.
Oh.
Impressionist
Hey.
oceanography.
Oh, we're filtering.
Yeah.
Oh man, so much wordplay on that.
Wow. Oh man, it's like a triple.
That was
Next
super.
level.
Oh.
Oh.
I gotta leave.
See ya.
There is a very distinct line though when it,
Yeah.
when it
That is-
clears.
Oh,
Yeah, that
if you
is
look
weird.
actually down and to the right, it looks like there's another one below a limpet that might have the same feature.
Do the polynoid scales kind of thicken over its lifespan? Like, in an older adult will have a thicker scale than
to be honest, I don't know. I know different species of them. Ones that I've sampled that have not been associated with hydrothermal vents are a lot larger, like some in the Antarctic, and they have really thick, leathery scales. But yeah, I don't know if that's a function of age. I'm not entirely sure.
And by thick and leathery, I have to ask Lee, are these squishy or crunchy?
Squishy.
Squishy.
Are you gonna lick it, Sam?
No. No, but I think that might have been discussed earlier because there is a comment to that effect.
Oh yeah, it's really diagnostic, especially with
is.
the acoustics. you know.
Exactly.
See now,
Crunchy then a squishy on the interior. Yeah.
if you look at that one under the probe, the stripe has disappeared. So I
leave yours off
think
for
it's
now.
just a function of the light on the scale patterns.
I think you're right. There's two sets.
Oh,
There's
wow.
a pair of them. You're absolutely right. So yeah, maybe it was just the angle we were looking
so
at, and it was
interesting.
it's just coming back into shot at the back
Huh.
towards the dorsal end.
Oh, that's
But-
super interesting.
Yeah, that is interesting.
Or it's trying to signal to us.
Mhm.
Interesting. Good eye. Oh.
Ah, here it comes, starting to grow again.
Oh.
We got a temperature update. Where are we at?
16.
1.6.
Yes,
16.
correct.
Thank you.
Do the scale worms have any sort of internal organs or fluids that would be moving through them that could be creating that effect as well?
That I do not know. I do not know.
'Cause I've seen that with other types of worms, but
Now that Jess pointed out, I wonder if it's at the angle where you get the maximum light penetration through the layer of the scale. If there's some transparency in that, the thickness of penetrating it.
Yeah.
Yeah, I put in a ship move.
Or to Sam's effect, I wonder if they are opening
210.
35.
opening their scales and closing them,
Copy that. 35.
depending on the environment and that moment.
can they
Oh, sorry.
can they open their scales, Lee?
No, I don't know,
No.
but I think it might be a function of actually as it's
Okay, let's
crawling
turn
over
yours on
rough
again.
terrain that actually the scales are like spreading apart at the top potentially. Maybe that's what's going on. Who knows?
Well, we've got, what, another 20 minutes to study this little guy?
Sorry, I just started it again. I popped that widget up.
So viewers wondering whether it's the temperature of the vent, the gases, or the flow rate that is causing this wavy shimmeriness in the water, which is a great question.
Probably a combination of all of that, I would think.
think it's
think
mostly
so.
the temperature differential.
Correct,
Yeah.
yeah. It's primarily the temperature.
It's
So
like
we
when
think
you see a mirage,
absolutely like on
Top
a
of a
hot road.
barbecue.
top of a barbecue. Yeah.
Just
Well,
clear
when
end.
you look at like a hot asphalt, you get ray bending at that interface. So sometimes you can get an inversion, so the ground appeals above the interface because it's bending up.
Those are a lot of big words, Ben. Can you break that down into simple terms?
The light bends as there's a temperature difference at an interface or across the gradient of temperature.
Hot stuff looks wiggly.
Thank you, Trevor.
Roger.
Hey, guys.
Hello.
Hey, I think we should stop this sample and move on to the
Okay.
next filter.
Okay. Copy.
That
Copy
was
that,
about
Judy.
15 minutes, 20 seconds.
Okay, stopping sample 1956.
Okay, we got a 0.28 liters per minute flow rate.
0.28 liters per minute.
Volume was 5.65 liters.
5.65 liters. Copy that.
Moving to port 3.
Hmm?
Copy, moving to port
You
3.
didn't say it.
And start.
Copy that, starting sample 1956.
Well, it started fast.
There you go.
Slow down.
Oh.
Can you line
What
this
is
up a
going
little
on with
better?
filter
I
two?
bet
See
there
the
was a
tether?
hole in it.
In that one, or like the filter wasn't lined up or something?
Yeah.
Just make a note of it and look at the filter when you
Okay.
guys
Yeah.
open them.
Okay. Ugh. It's painful to watch.
It is, isn't it? It's like,
Yeah.
oh no. But reassuring at the same time for you guys now. Now you
Yeah.
have two that are doing the same thing, I
Yeah.
suppose.
At least it's moving.
It's moving. Do we have a temperature?
33.
33, thank you.
Forgot to write down this.
There goes the shrimp.
What do you think our high was for the last filter? 52? Does that every time.
Hey guys, can I take eyes off of the flow meter for a second?
Sure.
Thanks.
Lee, can you see, and I think it's 7. Do you see all that stuff floating
Oh
around?
yeah. Oh yeah. Oh, is
You
that
think
what
that's
it's?
shrimp?
Yeah.
Cool.
Oh. I'm actually just having a quick look to see what they are because, yeah, I've not seen that event before. Not like tiny ones like that.
Yeah.
Couldn't it be zooplankton?
It could be.
Maybe.
You could be right, Judy. I have no idea what it is. But it's...
You'll see them every once in
Yeah.
a while go in front of the flow meter or like up in the Argus camera. There's a few maybe.
Okay, I'll keep my eyes peeled.
It's hard to see, yeah.
No, but I can
But
see
they
what you're talking
definitely
about
looked
now.
orange to me, and
Okay.
that made me think they were shrimp, but they could be something else.
Lee, do
Yeah,
you
I think they're
know?
juveniles or something?
Do you know if anyone's built like a stainless steel plankton net
29 degrees.
to?
Stainless steel plankton net.
Yeah, that we could, go
Oh,
through
like,
that.
actually, like physics. No, that's one of the hardest things is actually trying to like
Capture something like
sample,
that?
yeah, any kind of planktonic
Okay.
larvae or any small zooplankton
Yeah.
at depth. I mean, similar to what you're doing as well. They've
Yeah.
put like SAPs, like standalone pumps,
Yeah.
on ROVs,
Just to-
and then
just
with
to-
a larger mesh filter,
Uh-huh.
and then just tried to like filter the water.
Yeah,
with
they have to
that.
they have to filter so much.
Yeah, but these guys are quite tiny. Not in my field of expertise.
Yeah.
But no, it is interesting, and to be able to see them on that camera 7, that's incredible, actually. There's a lot going on in that camera, actually. There's loads of like
Yes.
little,
It's like a little plankton party.
yeah, little bits of vigorous flow or something, and then a load of. That's interesting. Huh.
Wait, where's that one pointed to?
The
7?
boxes are on the back.
Oh.
Cam 7, that's Herc
I just saw
starboard
one.
camera.
That's those are the filters
Starboard.
on the starboard side. Got it.
Oh, that's the filters?
Yeah. See the
Okay.
box and
Oh,
then
yeah.
the tube
I thought it was the
areas.
Yeah, so we're looking directly. If you look at the Argus cam, you can see exactly we're looking straight at that. At your
Oh,
vent,
I
the
see, I
vent
see.
chimneys from the
Okay.
candelabra
Yeah.
side.
So it's kind of at the base of that
Yeah.
chimney. We should do a drive-through. Whoa! Look, there's like a plume.
42 degrees.
Yeah, you get those little eruptive kind of moments. Look, I don't know whether that's a function of actually our thrusters are maybe generating that, because we're quite close
Oh,
to
yeah.
it. So if we're trying to stabilize a vehicle on site, then
Thrusters
maybe
have been constant.
They
There's
have-
no
oh, they're
no
constant.
changes. Yeah.
Oh,
Oh.
interesting. Then that is weird.
That's neat.
There's like a little
Looks like little
vortex.
tornadoes.
Yeah, you can see it. It's like a little vortex, like spinning up.
Do you want me to throw that on sat feed 2 for other people to see?
Can do, maybe that's just Argus, isn't it?
Yeah.
Yeah, you can do that as long as that doesn't affect anyone at ISC.
Just Argus?
Sorry.
Oh, Lee, ouch.
Ouch. Sorry, Cassidy.
That
Sorry,
hurt my
sorry, everyone.
feelings.
Argus is looking
I
good
thought we
too.
were a team.
Argus
now
is looking
everyone's
good.
now everyone gets a chance to see this little plankton party we're trying to figure
Plankton
out.
party.
I'm gonna remember that, Lee.
I know, I feel really terrible. I'm sorry.
You should feel terrible.
They're all coming to see the show at the candelabra.
That just sounds like the most fancy show in the deep sea.
They all have little bow ties on.
Oh, little top hats.
Yeah.
Welcome to the
Did you guys really see a
candelabra.
top hat?
What?
Did you really see a top hat?
You don't? You don't?
You get a temperature update
I
quickly?
forgot to put the flows. I'm assuming it's the same.
It is just the same from previous, right?
Yeah,
Where
I
the
think
end part
so.
is? Okay.
Sorry, I missed that. Sorry, was there a temperature update? What?
Oh.
Sorry.
Oh, no, we were talking about something I forgot to do. Temperature's 36.
36. Oh, it's pretty good. It's been pretty consistent then, hasn't it? It's not really dropped down too much.
Yeah. It's a good vent.
Yes.
Venti McVent face.
No. That one didn't make the short
That's
list,
a
did
great. Yes.
3 times.
Yes.
It was.
Oh, really?
Yeah.
I don't think Julie. I don't think Julie likes it.
No.
it? I'm with Julie on that one.
I
I
don't
know.
really like it either.
What don't I like?
Venti McVent face.
I am a big fan of that name.
Should we take a vote?
No. These votes always go wrong.
What about
Talking from experience. Don't do it.
what was the other option besides Venti McVent face?
Venti
we have
McVent
one
Field.
more that we're saving for tomorrow, which is the pièce de résistance.
Ah, the perfect name.
Yeah.
Contest winner.
Oh,
Yeah.
do you?
Oh.
Ooh.
It was
Oh,
a
I'm excited
blend
about that
between
one.
my 5-year-old and Eric.
I think a 5-year-old should win.
No, it was so So,
close.
should I tell them what it is?
Did you put it out to the vet? No, I feel like you should hold
Leave it
it.
a surprise.
Big
Yeah.
reveal.
We'll build it up.
Yeah.
We'll tweet about it.
I still think someone should have used Ace Ventura.
It's the perfect one. Yeah, and I wanted to use Adventure Time too.
Oh, that'd be cute.
Yeah,
Do you want?
but it didn't make the cut.
You want one?
In the early AM, we had a lot of questions from the audience that wanted to know where they can follow the information on the cones, on the orange cones. So I was telling them that Woods Hole Oceanographic Institute is doing the research on that, and so
We're at 67 degrees. Sorry.
no. So if Drs. Who and Dr. Smith would be willing at some point when there's time to share the Twitter account where people who are following your research can find the information.
Yeah. Yeah, you can, our Twitter handles are somewhere.
Yes.
You can share our Twitter accounts for sure.
Okay, thank you, doctors.
Doctors.
Yes, that's what you are. PhDs, you worked hard for it. Who and Smith? Who, Smith and Marsh?
Guys sound like a TV show.
Mm-hmm.
Next season.
What did they find at the vents?
Find out tomorrow.
Temperature's 59.
Copy that. 59.
We have 10 minutes left.
Both Cassidy and Trevor have been really good at turning these dives into action thrillers.
You should just wait for the sequel.
So how many more vent names do you actually have ready to go? You only have one? Oh, sorry, wrong question, wrong time.
We have a few.
You got a few?
Yeah. Do you want to submit one?
No, I'm just intrigued. I want to know how many sites you got left and how many might come out.
Amy's the mastermind.
Ha ha.
Yes, I was eating a Snickers.
Sorry.
Sorry.
No, sorry. I realized as soon as I asked that question, I was like, oh, no, wrong time.
Yeah, so we chose like 5 names out of 30.
Mm-hmm.
And the last name is tomorrow, I think, right?
I believe so.
Yeah, but we have the option for more. Like we had Venti Latte. And we had
Venti
the option-
That might
Latte.
not get used.
No,
We
it got
did.
used yesterday.
We
Yeah.
did.
Yeah.
And then,
Or
but we
day
had also like soy Venti Latte and mocha latte, no whip. All
a
of
whole
those kinds of
theme.
things. Yeah.
Whole coffee theme.
Yeah. See, vent.
Well,
venti
Amy,
latte.
we do have to name the octopus plate.
Oh.
Yeah. I can think of something really good for that.
Yeah?
I did have
Okay.
a whole bunch of like s'mores and toasted marshmallows and pink bubblegum unicorn land, you know, that kind of stuff.
I see. I understand the theme.
Well, Julie had
So it's
said
all
something
from
about
your kid?
marshmallows. Yeah. You'd mentioned marshmallows before we left, and so I got on this marshmallow theme because all of the vents are kind of black and white, and they look like toasted marshmallows in a way.
Oh, they
So I
do,
had a bunch
yeah.
of like those
Well-
kind of themes,
Yeah,
campfire.
there's a marshmallow event at Axial Seamount.
Oh, there is?
Oh, is that?
nice. You also
Maybe we
packed,
should call this one Shrimp Party.
you also packed
Yeah.
marshmallows. Like we opened our
I
gear up and you
did.
had surprise marshmallows
I
from
did,
past
'cause
Amy.
I forgot. Yeah, like I ever eat marshmallows, but I just decided I had to bring marshmallows on this trip. I've eaten one and I almost threw up. It was gross.
What
It
happens
was like
when
a
there's
strawberry
Jess, can you zoom out,
flavored
please?
one. Yeah. I thought it was going to be good, but it was too sweet.
So there's also at Axial Casper
Oh.
and Virgin. So there's a bunch of white and hydrite chimneys, and they've all been named different things.
Oh, cool.
Ah.
Look at that fish over there.
Actually, I got on a. Oh wow, that's a big fish. I got on a
That's a big
Ghostbusters kick
That's
too
yummy.
because I was thinking Stay Puft
Seaman
Marshmallow
Argus too, yeah.
Man, and then I had all these Ghostbuster themed ones, and
Yeah.
I thought it would go well with the Purple Rain in 1980s.
Yeah,
you know?
the 80s theme. I like that one.
All right, Jess, you can zoom in again,
Spent
please.
way too much time thinking about vent names. But most of the names that we chose were submitted by people we had a little contest, Sarah and I, on Twitter called Name That Vent. And so we had some people from the public would submit names so they could name the vents.